Hello everyone, my name is Fayaz Khan Sahil and today we're gonna learn about enzyme. So before learning about enzyme, let's start with what is a catalyst. A catalyst is a substance that increases the rate of chemical reaction without being changed or used in the process. It does this by lowering the activation energy. So, what is the activation energy? Activation energy is a minimum amount of energy required for the reaction to take place. Let's look at a diagram to understand activation energy in more details. As you can see in the y-axis, we have free energy or energy. It's the reaction coordinate in the x-axis. This is actually the activation energy. This is the energy required for the reactant to go into the transition state where they are converted into products. Now let's move on to our main topic. What is a enzyme, right? What are enzymes? Enzymes are proteins produced by living cells that act as a biological catalyst. So, what is enzyme made of? It is made up of proteins. Different enzymes is made up of different proteins. What they does is that they speed up the rate of chemical reaction without being used up in the process. What does this line actually mean? For example, an enzyme that look like this before starting of the reaction, at the end of the reaction it will still remain the same at the ending, right? So there is no change in their shape or structure, right? They are found in all living organisms, including human. In our stomach, we have pepsin, trypsin. We have a lot of enzymes in our own human cells, and we also found enzymes in plant, animals, fungi, bacteria, and play a vital role in all metabolic processes. You know, all metabolic processes in our body is carried out by enzyme. So enzymes are fundamentally one of the most important part of our biological system. Let's look at the structure of an enzyme. As you can see, this structure, let me draw it in different pen. This structure is the active site, okay? And this is a substrate. If you look closely, you can see they are complementary in shape, right? They can fit with one another, right? So let's look at how enzyme actually work. Enzyme has an active site. So this is the active site. So in this active site, this substrate actually bind to form enzyme substrate complex. In the next diagram, you can see it very carefully. As you can see, the substrate and the enzyme have a complementary in shape. So they fit with one another, right? So this is known as enzyme substrate complex. Okay? So what happened after enzyme substrate complex? Let's look at it. As you can see, this is another substrate. This is another substrate. It bind to an enzyme. It form enzyme substrate complex. Then as you can see, these bonds get broken down to release two products, right? So the function of an enzyme is very easy, right? It convert reactant or substrate into product. That's their only job, to convert substrate into product. In O-level exam, they may give you to draw this diagram, so read this thing very carefully. Now let's look at one of the important concept, lock and key hypothesis. Interestingly, when I was in O-level, which is about 6 years, I guess, 6 years ago, when I was giving my O-level, I was always confused about lock and key hypothesis, which is lock and which is key in the enzyme system, right? Let's start lock and key hypothesis. The lock and key hypothesis explain how enzyme work. Each enzyme has an active site, right? Suppose this is an enzyme. Now this is very bad drawing. Sorry, your sir is very bad at drawing, but stay with me. Don't give up. And I'm a bit nervous. I'm really new to the YouTube thing, so just bear with me. By the way, write down in the comment section where you guys are from. I would love to know you guys in more details. Enzyme, and this is substrate, right? As you can see, enzyme has an active site, right? It has an active site. Oh my god, my handwriting is so bad. Active site, okay? As you can see, the lock and key hypothesis in the text, the enzyme has an active site with a specific shape. This has a specific shape, right? And the substrate fit exactly into the active site, just like a key. An enzyme act as a lock, right? When they fit, the reaction happen. When this fit, the reaction happen and products are released. Simple concept. So, which one is key? The substrate is actually the key. Like it's just fit into the active site, and the enzyme is actually the lock. So don't forget this thing. In lock and key hypothesis, the substrate fit exactly into the active site just like a key, and enzyme act as a lock. So never mess up this concept. In this diagram, you will be more cleared. As you can see, the key is the substrate and this is a lock. Lock is the enzyme, then enzyme substrate complex form over here. In enzymatic diagram, you can see the substrate, the enzyme active site. It binds to the active site and form enzyme substrate complex. So when they tell you to write in paper 2, how lock and key hypothesis work, always mention the key. The key is the substrate, lock is the enzyme, then enzyme substrate complex. These actually hold 1 mark. This actually hold 1 mark in 3-mark question. Okay, I think let me erase this thing. Let me draw it more carefully. These actually hold 1 mark for a 3-mark question. Okay, for example, you need to mention key is the substrate, lock is the enzyme. This is 1 mark and this is 1 mark, and how they bind to one another, this is another mark. Okay, now this is one of the most interesting, and most probably it comes a lot, the effect of temperature on enzyme. If you look closely at the diagram, we have in the y-axis we have rate of enzyme activity, and in the x-axis we have temperature, right? Let's look at the diagram. Okay, as temperature increases, enzyme molecule and substrate gain more kinetic energy, so more number of collision take place. As the temperature increases, the rate of reaction start to increases, right? Until it reaches a temperature which is known as the optimum temperature, where the enzyme works the fastest. And after optimum temperature, you can see a desired trend. The rate of enzymatic activity started to decrease actually. This is because as the temperature increases, the active site, right? For example, the enzyme active site, let's draw it over here. Enzyme active site, it actually hold by different type of bond. For example, hydrogen bond, covalent bond, disulfide bond, right? So at high temperature, these bonds started to break down. As a result, the active site started to change its shape, right? So if the active site changes its shape, the substrate can no longer binds to it, right? As a result, more and more enzyme begin to lose its active site shape. So as you can see, the rate of reaction continue to decrease as less enzyme substrate actually forms until and the rate of enzyme activity decreases until you can see denaturation point, where no enzyme or none of the enzyme can actually binds to the substrate. So it can no longer bind to the substance. Once denatured, enzymes stop working, the reaction slow down or stop completely, right? As you can see, after the optimum temperature, as the temperature increases, I'm summarizing it, the enzyme slowly started to lose its active site, so less number of substrate could binds to it, right? As a result, the rate of reaction started to decrease. The same concept actually applied to the effect of pH. As you can see, change in pH significantly impact the shape of an enzyme active site. In the graph, we can see as the pH increases, the rate of reaction actually increases until it reaches its optimum pH. Different enzymes have different optimum pH. For example, enzyme pepsin work in our stomach that has an optimum pH of 2.5, right, which is here. And as you can see, for this enzyme, they have no activity at this pH, right? However, at pH 8, which is slightly alkaline, the enzyme has the optimum pH and the enzyme work the fastest actually. However, further increase in pH causes a sharp decline because enzyme active site loses its shape and becoming denatured and ineffective. Now let's move on to the last slide of our lecture, which is limiting factor. So what does this whole diagram mean? In the y-axis we have rate of enzyme activity and in the x-axis we have substrate activity. Enzyme work efficiently when there is an ample supply of substrate, which simply means enzyme activity increases if there is more number of substrate available. So more number of enzyme substrate complex could be formed. As the substrate concentration increases, right, the rate of enzymatic activity also increases. As you can see, the rate of enzymatic activity also increases until it reaches this point. This is known as enzyme saturation point. At this point, let me write it for you guys to be more clear. Enzyme saturation point. So what happened in enzyme saturation point? In enzyme saturation point, all the enzyme active site is filled up with substrate, so no new substrate could bind to it. So the reaction actually reaches a plateau, right? As all the enzyme active site are being occupied. This is known as enzyme saturation point, where adding more substance won't increases the rate because all the active site are already in use. So we are done with our today's lecture. Please stay tuned and do subscribe to my channel. It will mean a lot to me, and bye bye. See you guys in the next video.